warm welcome to everyone and a very good morning to everyone present here. I am Ruxana Pradhan, a research scholar from the Department of Zoology Utkal University. Welcome you to the second day of International Conference on Technological Innovation in Animal Science Research and Social Transformation. Before we begin our program, I want to make a small announcement. The evaluation for poster presentation PP 41 to PP 60 shall be done from 11 a.m. to 12 noon, and the evaluation for PP 60 to PP 80 shall be done from 12 noon to 1 p.m. Let's begin with the technical session, and let's invite our first plenary talk. May I now request Professor Sarvani Giriman, Dean, Har Govind Khurana School of Life Sciences, Assam University, Silchar, to chair the session. Thank you, ma'am. Let's invite our first plenary speaker, Dr. Subrat Kumar Das, scientist at Institute of Life Sciences, Bhubaneswar. His research area is microbial ecology, genomics, and translational research. He is a PhD and postdoctoral fellow, Department of Microbiology and Immunology, University of Texas Medical Branch, Galveston, Texas, US. To his credits, he has developed wholesale livestock vaccine for respiratory diseases. He has contributed book chapters in Berkeley's Manual of Scientific Bacteriology, the Williams and Wilkins Co. Baltimore. 25 new bacteria have been reported from his laboratory. He has edited 2 books in Springer and Nature, Volume 1, Microbial Diversity in Normal and Extreme Environments, Volume 2, Microbial Diversity in Soil and Agro Ecosystem. He has received E.K. Janaki Ammal National Award for the year 2015 from Ministry of Environment, Forest and Climate Change, Government of India. He has received Professor K.S. Bilgrami Memorial Award from Society for Plant Research, India. And today his title of the talk will be Exploration of Microbial Resources and Translational Research.
Good morning. Okay, good morning. First of all, I'd like to thank to the organizer, Professor Prabhati Madam, Professor Mahapatra, that is Prabhati Mahapatra of Department of Geology, and the other faculty members, Professor Dr. Sahu, Dr. Sujoko Ponda, and other students, staff of the Department of Zoology to organize this international conference and giving me the opportunity to share some of my students what they are working in my laboratory at Institute of Life Sciences, Bhubaneswar. And I am very much delighted to see that Professor Luiten over here, and he just long way from Belgium, he came to attend this conference and share his idea, research, and the in the present time of research that drug discovery from the different pharmaceutical products, from the traditional plants and other natural resources. So with these few words, I will start my lecture that exploration of microbial resource and translational research. So last 20 years I am at institute. Just then when I came here, that I thought that there are potential to explore the natural resource, that especially the microbes, that for the next generation drug discovery. And last 10 years I spent that only microbes associated with the corals from the Andaman Sea and the total eastern part of the country to see that extremophiles, that is the microbes living in the hot springs. So with this, actually some of the potential microbes we have identified, characterized, evaluated their potential role as a pharmaceutical, and one of such microorganisms I have developed and characterized that can be used as a to control the respiratory disease, that is the livestock, wholesale livestock vaccine for the respiratory diseases. And besides some of the work we have extended in the metagenomic research that was initially started in the University of Wisconsin, that Professor Joe Holderson. Then subsequently, almost after 15 years, that approach has entered into our country, India. There's a lot of national laboratories they are trying to contribute. So the first few slides, I will just give some overall thematic approach related to this topic, and then I will just describe some of the things that the area of research that microbial ecology, that phylogenomics and functional characterization of microbes and microbiome, then microbial genomics and translational research. So just last week, yes, just I am confined with these two objectives only, but I am not never deviated from any other area. So that the novelty and the importance that microbial resource in natural ecosystems still are explored, and metagenomic study can facilitate better understanding on uncultured microbial resource in any ecosystem. And studies on microbial diversity can help for the exploration of novel genes, biomolecules, and next generation pharmaceutical or the drug discovery. So research question, that what question we are going to address, that identification of new microbes from extreme environment in this qualitative approach, composition and functional characterization of microbiome by metagenomic approach, many microbes in very pristine environment and the ecosystem, we are not able to cultivate them in the laboratory condition. So there are so many, almost more than only 1% microorganisms we can cultivate in the laboratory, and remaining 99% is unculturable, because in the laboratory condition, whatever the medium or the media we need to improvise, or that because natural environment is a composite and the complex and the compound substrate. So that substrate or the identification of particular ingredients, we are not able to provide them, and many of them are the anaerobic in nature. So when the cultures are anaerobic in nature, their generation time is too prolonged. So that use those microorganisms in day-to-day, like E. coli, you have the generation time is only the 20 minutes. But the other microbes, anaerobic, either methylotrophs, acetogens, or acetogenic bacteria, or methanogenic bacteria, and other anaerobic bacteria, they contribute tremendously in the biogeochemical cycle in the environment. But those things is very difficult to cultivate in the laboratory. So it is not like normal mesophilic bacteria or normal bacteria, or like that. So and elucidation of the pathway and identification of genes encoding the sequences of novel biomolecules and the bioproducts. It is very interesting nowadays. Although when the alchemist first introduced the chemical composition, then subsequently a lot of metabolic pathways, the main focus was the biochemistry, and almost all major basic contribution has come up from with the study of microbial biochemistry and the physiology, genetics, and subsequently from prokaryotes to the approach has been shifted to the eukaryotic model system. And now the cell culture facility, people are trying to address that which drugs target site, so and so forth. So when people are talking about the microbes, they have to select the particular niche. It is not like that one can do each and everything, whatever the things are coming in their mind. So the niche, what niche we are going to address. First, it could be any pristine environment or any ecosystem. Pristine because where there is no anthropogenic activity, there is no invasion of the other human beings, the natural ecosystem, untouched, or the vaccine ecosystem, that people can look into it, that they can get some new microbes with their new potential or the biotechnological application. Extreme environment conditions such as hot springs and marine source, hydrothermal vents, cyclophiles, and the halophiles, barophiles, that eusteria, very unique ecosystem. But a lot of biogeochemical cycle with the changing the oligotrophic environment of the ecosystem. Any oligotrophic dynamics or the population dynamics is more abundant in the estuary. That why? Because a lot of that sweet water or the normal water and the marine water, it is a combination of these two, so that there are a lot of new type of habitat or the new type of microorganisms, new type of animals, sea animals or the flora fauna one can look into it that the history. The animal got microbiota at different trophic levels. You know that the omnivorous, carnivorous, and the herbivorous. So irrespective to human or the animals, you will get a lot of, because it is the basic composition is the carbohydrate, protein, fat. So when the carbohydrate, protein, fat, then the herbivorous, they mainly that depends on the that their gut will be mostly abundant with the bacteria that is degrade cellulase or the starch, cellulase, hemicellulase, and other sugar and other things. Like this, that carnivorous, they're mostly the protein-rich food they consume, and the herbivorous and the omnivorous, where you can get both the combination. That some of the signature organisms you can get that identify or they are mostly abundant for the degradation of the protein compound and protein foodstuffs, and some of the like meat and other like fish, meat, whatever people are consuming. The elucidation of microbiota in their gut and their role in carbohydrate, protein, fat, that is food habits. That also tremendously change. Then microbial population dynamics compared to disease and healthy individuals. Lot of paper, lot of research nowadays, global people are doing all over the world. That obese, non-obese, then the person having the normal buccal cavity, the flora and the person's disease with the buccal cancer or the oral cancer. Then how the microbial flora fauna change. So that flora change, that is also one of the promising area now. That microbial population plays a huge role for the disease progression and they are causing the disease, so and so forth. Microbial population dynamics is changing oligotrophic environment. Oligotrophic environment is very short here. Even January to December, if someone can, monitor time to time, they will see that how the oligotrophic environment change with, the variation or the seasonal variation. Then one control of microbes causing disease in human, animals, and the plants. And also one can look into it how cultivation, like that, crop rotation, how the particular agricultural field, when the legumes you are growing, what sort of microbial flora or the microbes are abundant when you are growing in the legume plants. But followed by some of the rice, then you can see that a lot of methanogenic bacteria, global warming, because a lot of methanogenic bacteria generally they grow in the rice field. But generally, in the high-yielding variety rice, generally need 3 to 4 irrigation throughout their 3 months period. But in China, they reduce the number of irrigation. But in our country, if we just reduce that one, instead of 3 or 4 irrigation, if we can irrigate only the 2 times, then a lot of methanogenic bacteria, they are generally love to remain in the, or they remain in the soil. And when they get the waterlogging condition, they emit the methane, and that methane is the greenhouse gas, ultimately leads to the global warming. And this study helps to understand the different biochemical cycle in the nature. So that is as a whole the works flow that if one like to investigate, they have to do first the 16 RNA sequence, but now the whole genome sequence is the thumb rule. That if someone is interested to do that, to contribute something or to either to analyze microbes, so that first you have to do the whole genome sequencing, and once you get the whole genome sequence, now you can analyze the entire genetic makeup of this organism. And then structural annotation, that is also bioinformatics expertise is required. That laboratory should have a person that who can look into the assembly of the gene. When the metagenome comes into the picture, that small fragment of 150 base pair nucleotide, how you can assemble in the 5 to 3' direction, 5' to 3' direction, so that you can reconstruct the genome. Because a lot of non-culturable organisms you cannot cultivate in the laboratory, but once you retrieve the genomic identity or the genome from the sediment or any gut, we do not know how many human gut in the human gut, how many archaeal population is there. then if the archaea is there, what physiological or the biochemical role they play in the gut, that is also not only human gut, other animals also. Annotation, structural annotation, you have to develop different pipelines, and many of you know that I am very much attached with the Joint Genome Institute USA, that I have a collaborative work with Joint Genome Institute. So the genomics part, a lot of analysis of the annotation sometimes whenever it requires, they helps me a lot. That laboratory should have one analytical group, that chemical analysis, that the chemical, so that one can understand the TLC, HPLC, then HPLC, HRMS. There are nowadays modern technology appear, NMR mass spectroscopy. So when someone is interested for the drug discovery, and when you will identify the compounds up to its homogeneity, so homogeneity part will come at the end, at least 98 to 99% purity is required when someone, and it should be soluble in the water. So sometimes people say that if particular functional group, if you change, that I have developed one compound also, that I am not anti-cancerous compound, that I am not going to present in this study from the microbial source. But it has a huge cytotoxic activity in the normal cancer cell line, the different cancer cell lines. So that structural elucidation and the identification, then how most of the natural compounds are the hydrophobic in nature. So when someone is interested to make it hydrophobic to hydrophilic, then the mode of action, then whether their structure will justify that, so that one can use in future. So there are so many, that chemistry point of view, that a lot of things one has to consider before think for FDA approval or our government of India, as a use as a drug candidate. So the next is the genomics, metagenomics. So it is very, in our country, I will say that 2020, 23, 24 onwards, that a lot of national laboratories, they start looking into that. We have to do the metagenomic study that initially started in the USA. 25 pharmaceutical company, that consortia, that is called the TIASRST. Almost all major, that is drug company in the USA, they start looking in the metagenomic study, taking into the sediment sample from the Tara Ocean to all Pacific Ocean, Atlantic Ocean, irrespective to that lot of, because the second generation drug discovery. Because the first, as we know the history, that Waxman, that first discovered the streptovirus, and his student Alexander Fleming, that he discovered that the penicillin. And then during First World War and the Second World War, and then after that, only the derivatives of the penicillin, that either aminoglycosidic group of antibiotics or beta-lactam group of antibiotics. That pharma people only, they do that particular functional group, they change it and they give different trade name and they occupy the market. But in true sense, the drug discovery, it is now very challenging, but a lot of people all over the world, they are trying to identify, they are trying to develop, they are trying to isolate the new drugs so that can use for the present time disease people are suffering, so and so. But different type of therapeutics, different type of other way to treatment the disease. So many medical science tremendously developed other approach so that many diseases can be controlled. And then second is that method, as I told, metagenomic approach can access almost all genetic resources. And then advances in sequencing and the computational biology have made it possible that otherwise the computational biology, without this, you cannot, that check-in program, there are so many new programs day by day is coming, where their bioinformaticians help how to organize the Watson-Crick model, the way they have done the phosphodiesterase linkage, then how you assemble the 5' to 3' direction, so that it can justify on complete bin or the genome, and with all the coding, non-coding sequences. As you know that eukaryotic system, that a lot of exon, that is introns, a lot of introns are there, but in the bacterial genome, the number of introns are very, very less. So, and the repetitive DNA sequence also very high in the eukaryotic model system. And many repetitive DNA sequence, only that few has been identified that they are really causing some disease or due to that mutation in the repetitive DNA sequence, they leads to develop some of the disease. Secondly, that and then when you will identify the niche, then first, as I told that, they are the oligotrophic environment. So these are one of the examples of the physical component analysis. But initially it was the mandate of the government of India, Department of Biotechnology. They asked me to explore the microbial resource in the eastern part of the country. That all 5-6 states I traveled a lot and collected the sample, including Odisha, West Bengal, and all Bihar, Jharkhand, Assam, then Sikkim, all northeast region I travel. And then any that physiochemical parameters, not only pH, temperature, alkalinity or acidity, but there are so many, that is ions, chloride ions, calcium, potassium, chlorine, bromine, fluorine, so that all you have to identify those who are very nearer to the coordinates, that whether those coordinates how they drives or they are acting as a driver or the driving force for the particular group of taxa or the microorganisms in that particular sample or the particular hospital. And second is that we identify different types of taxa, and these are the different types of genera. So that here, whatever I have presented, that almost all phyla and the genus, they mostly that they recognize the habitat of the thermal spring, and many of them are the thermophilic, many of them are thermophiles. They can tolerate above 50 degree, and many of them that representative taxa, they can tolerate above 70, 80 degree, and many of them archaea is there. As we know that most of the archaea are hyperthermophilic, starting from the nano. So, so that if the comparative analysis with the total database more than 25,000 16S RNA sequence, we consider to validate that our that unculturable reconstructed genome. And then this is the nowadays the problem, that is the resolution. That resolution of the tree or the phylogenetic depends on when the genome wide core gene, not only single 16S RNA sequence. You have to consider more genes and the housekeeping genes and other core genes, non-core genes. But core genes, if you take consider and do the phylogenetic analysis, then you can get that data that to identify two genome that based on the their total core genes and the 16S RNA. Then finally we have reconstructed and this article. And then each of them, if they either know mostly the carbon dioxide, and then we can, but ultimately this they enter into the acid and CoA, because acid and CoA have the urea cycle and the beta oxidation amination that is for the synthesis of a particular amino acid transamination reaction, so that particular keto group and the amine group, how they interchange each other, and all 20 essential amino acids. And this is the first report. They are not only first report from our country, actually that the nitrogen fixing bacteria. So that is a better genome. So it is a non-cultured bacteria, but it just reflects the genomic sequence, so elucidate them apart. So only that this much I will tell about the phylogenomic study of the single organisms to metagenome pool, how to retrieve that genome sequence from the unculturable source or the unculturable microorganisms. And it is showing only the 69% sequence similarity at the level of 16 RNA sequences, as it is thumb rule, that anything very closely to their taxon, it should be more than 97% sequence similarity. But we got the sequence similarity on the 69%, and other A9 and other data I have not presented in this presentation. But based on this, that we propose this organism as a new organism, unculturable Bachyarchaeota. So, second is that microbial genomics and translational research. This is my second part of the study, and that vaccine development. It is also many times I believe that if you think today that I will get the Nobel Prize, you may not get the Nobel Prize. If you think that I will publish this paper in the Nature Science, you may not be able to publish. That any commitment for but if you make yourself very committed that I will contribute to very good quality of science, I will develop or I will produce the data, artifact-free data, which can be validated all over the world. Any laboratory, if they use that methodology, this particular organism, so or any biomaterial. So whatever the data we have reproduced, the same data will be reproduced irrespective to country or the laboratory. So that it is also that anything when someone is achieved, it is achievement is a by chance. As we believe it, long 500 years back, that Newton, the way he just saw that apple fall. So like this, all the law of motion and other things, that theory that he developed. So based on that, it is also the by chance I got this organism from the not thermophilic bacteria, but it is a from a thermal spring here from here 172 kilometers in the Odisha state. And that when I was looking into mostly the biocatalysts, like industrially important enzymes like amylase, cellulase, lipase, gynase, so on and so forth. And other also that many pigment also having the pharmaceutical importance. Many bacteria, they produce a different type of pigment. So that pigment also can act as a pharmaceutical. So I got this bacteria, that Bordetella bronchoseptica, from the thermal spring. Then that I was thinking that Bordetella bronchoseptica, that strain Bordetella, as of today, almost all that is so far before the report is there in the literature, that they can grow only, they are host dependent. Extra host, their report is not there. What that people globally they isolated? Isolation was from the USA, and then subsequently from the Europe, from other part of the globe. But all organisms, either from the patient sample or from the avian, from the dog, from the horse, from the pig, so and so forth. So when I got this organism, then first initially we did the 16 RNA sequence, and I found that it is a Bordetella bronchoseptica. Then I became curious that how Bordetella bronchoseptica can live in the natural ecosystem. So then I started doing the the total. We have MOU and the material transfer agreement with the Indian Immunologicals Limited, so that I am going to transfer this technology, that the vaccine candidate, wholesale livestock vaccine, to our second largest Indian company, pharma company, the Indian Immunologicals. I am going to ceremony and then will transfer material. Then what is the bronchoseptica is an attenuated bacterial state. Then it does not produce the pertussis toxin. Due to many of our countries, they produce the pertussis for the whooping cough. So that whooping cough generally from the kids to age group up to 50, generally they immunize that not to have a whooping cough. But that Europe and USA, Africa, then Canada and Australia, all developed countries, they have a very fascination to keep 2, 3, 4, 5 pets in their home. So that most of the dog or the kennel cough, so they are suffering from the respiratory pneumonia or the bronchitis or the rhinitis. There are so many symptoms, and also in outside country that they are the piggery. There is a farming system, butchery. Then in pig also that, and also in the I have seen in the extensive in the USA that many people household they have the pet cat as a pet. from the lot of respiratory diseases. So they just take them to the veterinarian to immunize them. And the time to time, day one, then after 2 months, 1 month, then after 6 months, like human, papilloma virus, the way that our people take that girls are taking that papilloma virus, anti-cancerous cervical cancer. So that anti-cervical cancer vaccination. So like, and then additional genes encoding DARPA protein and then these are major virulent genes in the body level. Could be a better vaccine candidate. This bacteria that cannot revert to the vaccine phase as the above genes are absent in the genome. Therefore, we propose Bordetella bronchiolitica is a safer candidate for developing a livestock vaccine for the immunization of the livestock animals. So, rationale of using the Bordetella bronchiolitica as a candidate, Bordetella infection in livestock animals reduce weight loss. When that any animal that either carry that is dog or other hospital that they just reduce their body, and then more vaccine has function related to their effective immunoresponse. Many vaccination, but after the antibody titer acid, maybe that antibody titer is not that much. But a lot of people say we are immunizing the animal, immunizing the human, other animal. So what is the Bordetella particles? Actually, that Bordetella bronchiolitica is the evolutionary. I am not going to display those data actually how the Bordetella level of genome. So all genomic analysis we have done to show that Bordetella bronchiolitica is the more ancient, and then Bordetella pertussis and the Bordetella parapertussis has evolved, and subsequently as of today, 19 different species are there, and all these belongs to the taxa Bordetella, and all have more or less similarity with the ancestor strain that Bordetella effective for the 4 to 5 years. Now in the pharmaceutical single protein, the recombinant protein, they just immunize the express and they immunize and they see. But when you use the full vaccine, know that efficacy is 8. That is 8 to 12 years. Anything wrong? 8 to 12 years. This whole vaccine can be done are chemically detoxified for their side effects. But these organisms that since these genes are absent and some of the genes are mutated, so that normally people generally use to that inactivate them so that toxic effect with the vaccination process. It will not be required. And then still is to that do not produce these toxins, it could be the better vaccine candidate. So that if you look into it, that this strain we isolated from the thermal spring where the maximum temperature of that spring was 40 degrees, pH was 7.4, and these are the coordinates that the 72 kilometers in the, and the availability of the bacterial culture, it is available with a FTCC patent because we also in the foreign collection and it was isolated by me. All the molecular characterization has been done by me. So these are some of the physiological phenotypic things, and we to test it. This is a vaccine candidate. We procure one organism that has and all the study we used as a challenge strain to see that this organisms actually our organism giving the protection against the pathogenic strain. And Bordetella bronchiolitica, that is that. And total the genomic and ROD, we have total genomic map, we have run it, and we have seen that law of conservation of mass, law of conservation of. So that in the biology, that law of conservation, when that organism is gaining something, they will lose something. Because the law of conservation of mass at the level of genome, one has to justify. It is not like that the genome size is 5.2 kb, 2 MB, and after some time the genome could be the 5.8 kb. It is not possible. So there is a gene gain and the gene loss mechanism, that simultaneously occurred. So many like lateral gene transfer, new drug resistant phenotype among the microbes, because nowadays drug resistance in Europe is very much concerned about the new drug resistance. Many of you might be knowing that German people, they generally procure that salad from the Russia. Then that about 5 years back, that how the consuming the salad, that that E. coli infection was there, and probably 18 or 19 people, there is a casualty within 3 days people. Then within a day and so the whole genome sequence they have done, they found that new antibiotic resistance gene has been introduced into the acquired by that particular organism. And then doctor physicians has given the proper treatment so that they control that further dissemination or the contamination of the disease to the human population. So this is the total whole genome sequencing and the genomic architecture. And then everything we have done, and you should see that it is and it is showing that hello. Okay, it is showing that as I told before that Bordetella bronchitis it is trained. So this is the first identified organisms in the globe actually by European people. So they are making the cluster with this. And then we have seen the average nucleotide identity. These are the basic thumb rule. When someone is claiming that this bacteria could be this or that, then average nucleotide identity, if you see that our organism showing maximum similarity with the same USA strain that Bordetella, it was isolated from the human. So it is 99.51%, and bidirectional based heat, it is this many genes are they are that similar. And then we just total we analyze that what are the genes are present. So total that the regulation, the BBG, then how the toxin gene regulated in the Bordetella bronchiceptica. Then Parkinson's toxin not expressed, it is present. Gamma-necrotin is absent. Adenylyl cyclase hemolysis is absent. Then LPSO antigen, that is also absent. Then PIM2 is there but not expressed but present. So all genetic, and Partictin is there, but Partictin the sequence similarity only 69%. So based on that total genomic architecture, so based on that we just proceed further that we should look into it and we should try to develop as a vaccine candidate. So then the Parkinson's toxin, the total the promoter sequence here that that +1 that transcriptional start site, and here the A and the T, the Bordetella Parkinson's and Bordetella bronchiceptica, there is a point mutation is there. So that here if you see that Bordetella Parkinson's, they can express the Parkinson's toxin. Then our strain, another cell that is whole cell as well as that purified protein that is called that hit like that, and that particular it is not expressed that one. And we have also taken that the Bordetella bronchiceptica is 200 as a negative control, and our test one where that Parkinson's toxin not expressed. So here also we did not find anything. So then that total the genomic structure, adenylyl cyclase, here we just make a map actually H200 and RB50, it is a pathogenic strain. Here the total gene is totally absent in our organism. And then another gene is the adenylyl cyclase. A is the adenylyl cyclase and the gamma-necrotin genetic locus. Here also it is totally absent. So based on that, we have done the gamma-necrotin necrosis assay, and here you will see that organism infected with the that is H200 our isolate. There is no lesion, but Bordetella parapartitis and Bordetella bronchiceptica, or RB50. So they develop the lesion here. So yeah, it is also the safer that gamma-necrotin, the virulence factor, one of the not expressed. Another is the lipopolysaccharide 2 antigen protein genetic locus. So Bordetella bronchiceptica, here also if you see that this particular WLP genetic locus responsible for antigen, that is A band, is totally absent, and then other one is partly present here, RB50, Bordetella bronchiceptica, and Bordetella pertussis. So that hemoglobin assay also we have done the other virulence factor FAA, FIM2 and FIM3, and we found that in strain HD200 that FIM2 gene is absent or not expressed. So based on that, we have challenged the experiment using the RB50, and we used as a RB50 both the trachea and the lung after infection, 3 days, then 7 days, then 14 days, 21 days, 28 days, different time intervals. We just sacrificed the mice, and we have done the total animal model study, and then we found that yes, it developed the compared to the virulence gene and our strain, though they produce the sufficient level of antibody, that we have not done all 5 components, total IgG only, that tells that after 40 days of infection with the challenged organisms, this particular strain, there is no colonization of the pathogenic strain in the trachea or the lung of the palsy mice model system, and at the same time they develop the protective antibody. Then histopathological study we have done uninfected mice 7 days post inoculation, this one, and post inoculation Bordetella bronchiceptica, and the control and the Bordetella bronchiceptica we have not found any necrotic cells of the necrotic tissues as compared to the experimental one as a control without any infected HD200 strain. So besides also one has to look into it, what are the drug resistant genes are there. So these are all distribution method as per the guideline of the UK. So we have done that in a distribution method, and these are the resistant phenotype. But some of the drug resistant marker genes also we found when the genomic analysis we have done. So that BAC8 and the SARS2, that sulfonamide resistant and other resistant genes are there, and they are showing the bacillus resistant and the sulfonamide, the 98.95% and the 100% similar. And then we use that different type of that Bordetella, classical Bordetella. These are all generally the 4 Bordetella strain: Bordetella pertussis, Bordetella bronchiceptica, and Bordetella parapertussis. Then where our organisms actually stand. So again, based on the housekeeping gene, we found that our strain that initially isolated from the human, so it is showing the similarity or making a cluster with the HD200. And then total 52 virulence genes are there. We just generate the that where it can cluster actually that our organisms here also still HD200 with the USA that human isolate. So it is making the cluster, and remaining are the from different sources. The total genetic matrix, actually the percentage identity sequence divergence matrix, we have just elucidated this percent. There are huge genetic divergence are there, taking into the each and every, even the particles having the 5 subunit, also that subunit having the lot of variation at the genome level. Then when we use that 24 virulence gene related, that's showing the maximum not 99%, 98% below that. Then we found that 24 virulence related gene. Again, it is showing that Bordetella bronchiceptica with the human isolates. And challenge experiment in higher the dog and puppies or the piglets, we have not done it. Immunogenicity study we have done it, that this work will be done by the Indian Immunological Limited. How long it lasts, that we have not tested. Then storage condition, the cold chain, that we have not done. Then contaminants or impurities in the preparation, dose and length of treatment, because that is a canine bomb, it should be used as a vaccination for the dog or the puppy. So our country, the government of India guideline, we cannot use the higher animals for the experimental purpose. So So maximum we can go for rabbit or the mice model system. So we have done, but rest of the validation and the evaluation that will be done by the Indian Immunological Limited. And we have estimated only a total serum IgG, but other IgM, IgA or IgD and IgE, it will be that they will be treated. So here one slide actually I just forget to include it. That when we got it from the natural environment, then many people ask when we send this manuscript to Fence Microbiology later, they ask that you just give the evidence that there are so many environmental signature genes are there. So those somehow I don't think that how I keep that slide. But all 6 genomic islands we have characterized. So I show it here. All green. Somehow I missed that slide here. Here that all these portions, that is genomic island 1, genomic island 2, genomic island 3, the green color. It is that we are here and here. So 6 genomic islands are there, and those 6 genomic islands, they recognize the organisms that the particular gene function is required for their survivability in the natural ecosystem. And as compared with all the classical body strains, so these genomic islands or the genes are not there altogether. It is almost 420 every extra chromosomal gene has entered into that. And a lot of genomic regeneration has been occurs, and the redundancy that in the genome of ST200. So conclusion is that is the first reported environmental stage classical mammalian body genome sequence 6 accessory genomic islands. Conclusion is that the slide I somehow skip actually, and provided crucial driving force in evolution in the natural thermal space. It is a non-toxigenic strain. It efficiently colonizes the respiratory organ of bulk mice. Both infection-induced and heat-kill wholesale induced anti-H2 antibody provide effective protection against pathogen RB50. It is first isolated from the rabbit, and a lot of work in the USA, in the University of that is California. That Jeff Miller, he was the president of ASM, American Society for Microbiology, and a lot of work he has done actually in the partnerships, but not like animal challenge of the puppies of the kennel cock or the pig, so and so forth. And but Pasteur Institute is contributing a lot actually. I know because a lot of antibody I procured from the Pasteur Institute, and some antibody also I procured from the UK. So for some of the validation study, Western blot data to see that whether this really protein expressed or not expressed. So to study that. And it could be used for development of potential life or heat-kill wholesale vaccine for livestock immunization. So thank you to have with this, because this mostly people use as a the kennel cock or the pet like a dog. So that is with this. Once more I like to thank the organizer that they invited me to share this platform, share my research activity, what my students are producing in the laboratory. So with these few words, I will just end here. Yeah, of course.
Thank you very much for your excellent talk. I think it opens up a window in front of all of us to think about, and I really appreciate you for the. I really, as a personally, I feel very, I appreciate you because you have emphasized on the data reproducibility and ethics in research, and through your research, and publication, you have shown that how, science should be. So we have many questions, I am sure, but we'll take only 2 questions for your talk. Question from the organizer.
But I got your point that glacier mostly they are hydrophilic. But any if you monitor the temperature, mostly the ocean temperature during winter time, surface temperature, and the surface to below to the bottom, the temperature variation always big. Either Pacific Ocean, Atlantic Ocean, Indian Ocean, Bay of Bengal. So there is a surface temperature sometimes 28, then the little bottom to that surface, the temperature. So cyclophilic bacteria you can get in the ocean. But now that is the adaptive evolution concept is there. So when the adaptive evolution picture is coming, one organism over a period of time for its survivability or sustainability in a natural ecosystem, they will lose some gene, they can gain some, or they by this way they can sustain actually. But many cyclophilic bacteria has been reported from the ocean because the glacier melt, that bacteria is coming. But it is controlled now. Lot of disease actually that vaccine starting from that is pass through